Hello, everyone. I'm very happy to welcome you to this COVID-19 lecture by none other than Professor Stephen Holgate, who is a world-renowned scientist and has done extensive work in the field of asthma that has even driven the guideline development around the world. To briefly introduce Professor Holgate, Professor Holgate is the Medical Research Council Clinical Professor of Immunopharmacology at the Faculty of Medicine in Southampton. His is a name that is known everywhere and doesn't require introduction, but it's an honor to introduce him. It's an honor to have him on this webinar. And after completing his medical training, he was trained in Harvard, and from there he moved back to Southampton, and where he has led the research on asthma, various aspects of asthma, ranging from epidemiology to genetics, pathology, microbiology, immunology, pharmacology, and as I mentioned, that most of this work has led to the development of different guideline management strategies. Also, I would say that he's been a mentor to many of the leaders around the world. And to me, he has a special place in my heart and in my life, because he's been a mentor to me in many ways. And as well as I had the honor of working with him in WOW, in the World Allergy Organization, when he served as treasurer. He is the past president of the British Society of Allergy, as well as the British Thoracic Society. He's a member of the UK Natural Environment Research Council, special advisor to the Royal College of Physicians. He's on many trusts, as well as I would be most honored to say that over and above everything, he has been honored by the Queen with the CBE, that is the Queen Commander of Order of the British Empire, which is one of the highest honors that anyone can get in the UK for his clinical services. And most recently, he received the award of the American College of Allergy, Asthma, and Clinical Immunology. So it's my honor, Professor Holgate, to invite you to give this lecture, and over to you.
Thank you very much, Professor Pawankar, for that most kind introduction. It's a pleasure and a privilege for me to give this talk to your esteemed society, and I hope that the information I'm able to pass on will be of help to many of your members. This is a unique time, of course, as we have experienced over the last 3 months extraordinary circumstances across the world with this peculiar virus that really has affected everybody in one form or another, even though we may not have actually had the disease ourselves. So my talk today is going to concentrate on trying to understand the mechanisms whereby this coronavirus causes such havoc in the human, and then talk a little bit about some of the treatment and possibly what's going to happen going forwards. Before I do that, I need to declare some interests, and they're listed on this slide. And you'll see that I am a non-executive board director of a company that's making a treatment for COVID. I'm chairman of a charity that researched on anti-TNF, which I shall talk about again in a minute, and I do occasional consultancy work with pharmaceutical companies. So let me start at the beginning. I live in Southampton on the south coast of England, and I spent 1 year of my early doctoral training in a little town called Salisbury, which some of you will have heard of, with the Novichok problem that we had some years, 2 years ago now. But Salisbury is a very beautiful city with a cathedral, and you can see in the middle top part of this slide a painting by Constable of the cathedral, taken from Queen Elizabeth Park, looking over that lovely part of England. And in that town, there is a hospital, or there was a hospital called the Harvard Hospital, and you can see a yellow plane flying over it there. This was an American hospital used in the Second World War that after the war was handed over to the Medical Research Council to find to found the Common Cold Unit. And the Common Cold Research Unit is where the coronavirus was first discovered. June Almeida and Tiril Bino, working out of this unit, discovered an unusual series of viruses, first by electron microscopy and then by culturing them in epithelial cells that seem to have this peculiar corona shape. And you can see the electron microscopy picture here of this corona around the viral core. And this is June Almeida working here back in the early 1960s. So it's really amazing to think that only a few miles from where I live, this group of viruses were first discovered. But of course, that's not really what this story is about. It's about a group of viruses that have mutated and have become increasingly aggressive over the last few years. When I was working with Dr. Tiril in Salisbury, several viruses were known. These are those listed on the left-hand side here, which are known to cause the common cold, and that's why he was researching on them. But of course, in 2002, we had this dramatic appearance of SARS-CoV, which came out of China and affected a large number of people living firstly in China and then spreading around Southeast Asia, causing a lot of problems. And of course, Hong Kong in particular was affected during this period. Eventually that died down somewhat, and then in 2013, another related virus emerged called MERS-CoV, which was transmitted via camels and appeared in the parts of the Middle East, which really perplexed people until they realized that it was a virus related to the original SARS-CoV virus. And again, that caused a number of deaths, and there were cases that spread around the world. But it really wasn't until the end of last year that this dramatic appearance of COVID-19, the SARS-CoV-2 virus, caused this terrible disease that emerged firstly out of China and spread rapidly across the world. And as you'll see in a few minutes, we're just past 1 million cases in the world now, which is really quite frightening. And the whole story around this, of course, is that these viruses are gradually changing, mutating, and they're mutating as they escape from their parental animals where they are not causing a disease problem, mostly bats, to an intermediate host and then on to human beings. So this shows the history from the 30th of December when the virus was first reported in Hunan in China, and then as we come through the beginning of this year, we can see that various discoveries were made around the origin of the virus, what happened in Wuhan as a result of its severe effect on humans with lockdown, then a global health emergency declared by the World Health Organization, and then we get more detail about the pathobiology of this very strange virus, and in particular, how human beings become infected. In other words, the receptors through which the virus gains entry into the human. So this was a picture I took about 2 days ago from my computer, showing that there are now nearly 10 million cases of COVID-19 that have been described, and nearly 500,000 global deaths. And you can see in the center of this slide, the red dots illustrating the countries most affected by this virus as it spread from China to all parts of the world. Interestingly, there are still some parts of the world that are not affected, and you can see Australia in particular, it's really just around the East Coast, but also large parts of Canada and some parts of Russia are not yet affected. So the virus is still moving as I speak and spreading from one country to another. Well, I mentioned earlier on that the primary host for these viruses, these rather unpleasant viruses are the bat population. And it's been shown now that the bat population can harbor up to 400 different coronaviruses. And obviously the potential, therefore, for this particular species to spread more disease is quite profound. The very strange thing about all of this, as often happens with these virus pandemics and epidemics, is that there's an intermediate host. I shall speak a little bit more about that in a moment. But as we know, for influenza, pigs and poultry have been intermediate hosts for those particular respiratory viruses. So just as with them, it's highly likely that there's an intermediate host from the bat to the human. But as yet, we don't know exactly what that is. So on this slide, really, is shown the characteristic grouping of the different coronaviruses. And if I may demonstrate on the left-hand side here, you see this section here called 2b. This is the group of SARS-like viruses that have been identified and includes, of course, the Wuhan virus that we've just been talking about, but also several other SARS viruses, SARS-CoV, as you can see here. But as we move round, you can see that MERS-CoV is related, but a little bit distant from the SARS viruses that are currently spreading across the world. But then as we move across around this genetic tree of viruses, and we start to get out to these groups out here, these are the common cold viruses. So here we have human CoV229E, which is one of the viruses that David Tyrrell worked on. And you'll see genetically they're quite distant from these SARS viruses. Now, I did mention that there might well have been an intermediate host, and though it's not certain what that host is, this animal called the pangolin is thought possibly to be the intermediate host, and that the bat transferred the virus to this intermediate host, and then that transferred within these wet markets. The particular one referred to here is in Wuhan, to the humans who ate the relevant food. And of course, there's an awful lot of research going on at the moment to try and trace exactly what happened in the intermediate period between the virus leaving the bat and gaining access to humans. And I'm sure we're going to hear a lot more about that. So these are the various unpleasant coronaviruses that are causing or have caused respiratory illnesses. And it's a genetic map just showing the very minor changes that occurs from SARS-CoV, which is the one we're currently experiencing, SARS-CoV-2, which is the one we're currently experiencing, and SARS-CoV and MERS-CoV. And you see the genetic changes really are just the reordering of some of the genes, some of the splicing of some of the genes, some of the rearrangement in some of the genes, but really no major massive changes, just very subtle changes in the genetic makeup of these different viruses, which of course has increased their infectivity because SARS-CoV-2 is so much more contagious compared to the other two SARS viruses, SARS-CoV and MERS-CoV. So again, this is a world map showing the distribution of SARS-CoV-2, as the virus is now called. You see it's just getting into Africa, so there's a lot of concern now what's going to happen in Africa if it breaks out and spreads through. It's already in India, as you can see, and is spreading through India, and there's been major lockdowns going there, but probably the countries most severely affected have been the United Kingdom, have been Spain, Italy, and of course parts of South America, in particular Venezuela and Brazil. And these countries are now experiencing really very serious infections. And only this morning we're hearing that parts of the United States are having to close down again, particularly Texas and Florida, as a result of the virus spreading rapidly through those communities. So this is still on the move, and it's still causing very serious problems around the world. Now, the virus is unusual in that it gains access to the human lung via the angiotensin receptor and enzyme system. And this is a cartoon showing how angiotensinogen gets converted to angiotensin 1 by renin in the kidney. Angiotensin 1 then binds to the angiotensin converting enzyme, which splits it to form angiotensin 2. And angiotensin 2 then can activate several receptors, which can lead to, in the lung, edema, tissue damage, inflammation, fibrosis, thrombosis, and a variety of other unpleasant sequelae. Now, angiotensin 2 can also be split by another enzyme called ACE2 into angiotensin 1,7. And as you can see here, angiotensin 1,7 can bind to this receptor down here, which is MAS, which has the opposite effects to angiotensin 2. In other words, it protects against tissue damage, being antifibrotic, vasodilatation, and vascular protection. And it's this ACE2, the ACE2 enzyme, the angiotensin converting enzyme 2, which is the receptor for SARS-CoV-2. And you can see it binding there to ACE2. It can bind to the cell-bound ACE2, which enables it to be internalized, or else it can bind to soluble ACE2, because ACE2 itself can be cut by a metalloprotease called ADAM17, the same metalloprotease that generates tumor necrosis factor alpha. Well, let's get into the clinical work a little bit here. Some of the risk factors now associated with COVID-19. Here you can see the most powerful risk factor is age. This is mortality from the American CDC, and you can see that people over the age of 70 are at very major risk of dying of this disease, whereas youngsters down here are relatively incredibly protected. There are a variety of different other risk factors that have been found. This is a study only published a couple of days ago from the United Kingdom, looking at 17 million patients in the national health system in the UK, looking at the various risk factors. And as you can see, age is mentioned there, but also male hazard, being a male, uncontrolled diabetes, a variety of chronic medical conditions, including severe but not mild asthma, and importantly, ethnicity, particularly Black people and people of Asian ethnic origin seem to be at extra risk of this virus, possibly because of socioeconomic factors, but also because of the increased diabetes that tend to occur in this population. Children are amazing because they are almost completely protected from this virus, and there's a lot of speculation as to why that might be. One theory is that their ACE2, which is the receptor for this virus, is upregulated in the epithelium of the lung, and the type 2 pneumocytes that express this also have the ability to generate soluble ACE2 in the lining fluid of children's lungs. And of course, this soluble ACE2 could bind the virus and prevent it from gaining access to the epithelium. There's also ideas around trained immunity due to lots of virus infection in early childhood that might increase their innate immunity, and then that they also, young children also have a constitutionally elevated innate immune response in their lymphocytes, particularly the B and T lymphocytes and natural killer cells. So in other words, innate immunity seems to be probably quite an important part of why children seem to be relatively protected compared to adults. But of course, that has been also, children have also become quite a concern because they develop, in a very rare circumstances, a Kawasaki-like syndrome. It's been called pediatric inflammatory multisystem syndrome temporally. This PIMS-TS is described in the yellow box there, a variety of systemic symptoms, and is life-threatening for the very few children that experience this. So children on the one hand are really mostly very much protected, with the occasional child experiencing this very unpleasant Kawasaki-like disease, which can be life-threatening for them. Now, one of the things about COVID-19 is the fact that it produces a very profound systemic disease in certain people, whereas in others, it just causes a mild flu-like illness. And here is a description of some of the features of the systemic activity of this virus. If we just start with the lung, we know that the lung is the primary site of contact with the virus, though it probably first gains entry into the human body through the nasal epithelium. And it's interesting that loss of smell and taste is one of the early symptoms of infection with this virus. But then it moves down the bronchi, infecting epithelial cells, and gains access to the lung, where it can cause this very profound pneumonic syndrome, and then moves beyond the lung systemically, which is what I'm going to talk about in a moment. In the lung, it can generate a syndrome very similar in some respects to adult respiratory distress syndrome. One of the characteristic features of ARDS, of course, is this hyaline membrane shown by the red arrow there that forms around the alveolar ducts and the alveolar sacs. And this is, of course, one of the factors that limits oxygenation of the blood passing through the pulmonary capillaries adjacent to the alveoli. It can also cause an upper airway problem with inflammation. This is a part of a large airway with cartilage, and here you can see a bronchitis, the epithelium heavily infected with lots of neutrophils and inflammatory cells with this virus. So in many ways, it's resembling ARDS in passing through these different phases. The first phase being an exudative phase, which is inflammatory. The second being a proliferative phase, which is where this hyaline membrane gets deposited and fibrosis starts. And then the fibrotic phase is the last phase of the lung pathology of ARDS. And if we look at the two stages on the far extreme, it would seem that the early stage is really where the epithelium and the type 2 pneumocytes become infected with the virus. That leads to the recruitment of activated T cells and alveolar macrophages. And it's the alveolar macrophages and the monocytes with the T cells that become hyperactivated, and as you can see on the right here, lead to this vigorous inflammatory reaction in the alveoli, where the neutrophil is the dominant cell, along with monocytes and macrophages, and then the formation of this hyaline membrane around the alveoli, which reduces oxygenation of the pulmonary blood. And it's this transition from an inflammatory phase to this proliferative phase, which is the aggressive side of COVID-19 that seems to be the real problem. And here you've got an illustration of one or two possible ways that the disease might be controlled early on using interferons or antisense technology to block viral replication. And then later on, as I'm going to talk about in a minute, the various interventions that will intervene with the inflammatory cascade. A very strange feature of this disease, which is different from adult respiratory distress syndrome as it normally occurs, is the vascular changes in the lung. And here you can see on the left that there is thrombosis of small blood vessels in the lung here, and also microvascular leakage and hemorrhage into the lung as a result of breakdown of the capillaries that are underneath the alveoli, the pulmonary capillaries, with leakage of red cells into the tissue. This is a very strange local hemorrhagic reaction, probably induced by microinfarcts as a result of intravascular coagulation occurring within the microvessels of the pulmonary vasculature. And here we've illustrated here, both in the veins and in the arterioles, the fact that the thrombotic and hemorrhagic responses can spread out of the capillaries into these larger blood vessels, leading to pulmonary hypertension, which is one of the problems that these patients experience as their disease progresses over time. And in this lower diagram here, you can see the microhemorrhages that occur around the alveolar sacs, and the red cells that are then phagocytosed by macrophages, giving this very strange appearance of macrophages being stuffed full of partially damaged red blood cells. So this is a very unusual pulmonary response, and I don't think it's seen with any other lung disease. So in terms of the various mechanisms whereby SARS-CoV-2 actually gets into the epithelium and then produces its unpleasant effects in the lung and beyond, there seem to be some potential areas for being able to intervene. It uses a protein called the spike protein on its surface to bind to the ACE2 receptor, the angiotensin converting enzyme 2 receptor here. And this spike protein binds to that receptor, and as a result gains entry to the cell. But the very same receptor, of course, is the generator of this protective small peptide called Ang-17, which can bind to MASR receptors, which are anti-inflammatory. And as I was saying earlier on, that children have increased levels of the ACE2 and increased levels of Ang-17, which enable them probably to have this inhibitory pathway very amply expressed when they get infected with this virus, whereas as you get older, you lose ACE2, and therefore you lose this inhibitory pathway. So potential treatments, well, there's a vaccine being developed against the spike protein. There are ACE2 receptor blockers. There's the administration of soluble ACE2, and then there's this accessory enzyme called TMPRSS2, which facilitates the passage of the virus along with ACE2 into the cell. So this proteolytic enzyme, TMPRSS2, is also being targeted with an inhibitor. And in the yellow box there are the various therapeutics that are still in clinical trial. Of course, there are other ways of getting at this beyond interfering with the ACE2 pathway and the TMPRSS2 pathway, one of which is to actually block the inflammatory cascade with pro-inflammatory cytokine inhibitors or by mechanical filtration of the blood. And all of these are being tried in patients whose lives are being threatened by this terrible disease. But the real way of managing this disease, of course, is to develop a vaccine. And item 3 there on the right-hand side shows that the vaccine that is being, or the vaccines that are being developed are trying to arm the CD8 and natural killer cell population to enable the virus to be eliminated very effectively when patients are infected with it. We do not have a virus yet. Well, we know quite a lot about the inflammatory cascade in other diseases, especially ARDS, but also in many other chronic inflammatory diseases that has flare-ups. Here is a diagram showing rheumatoid arthritis, ankylosing spondylitis, psoriasis, Crohn's disease, ulcerative colitis, atopic dermatitis, going from right to left, and the various cytokines that are critical in terms of the disease pathobiology. And they become, of course, targets against which monoclonal antibodies or soluble receptors can be directed. So on the right-hand side here, we can see that the cytokines that are initially activated are the cytokines coming from the epithelial cells as a result of the virus infection. And these are the alarmins and other cytokines that bring in the immune cells for the first wave of the inflammatory response. And that first wave of the inflammatory response obviously, excuse me, I'll go back. The first wave of the inflammatory response involves this macrophage here and the polymorphonuclear leukocyte neutrophil. And the neutrophil is the dominant inflammatory tissue damaging cell in this disease. And then we have the T cells, of course. And then as it escapes from the lung and starts beginning to produce this very dramatic inflammatory response, we get a whole variety of new cytokines formed, which are the cytokines that are linked to hyperinflammation, or the so-called cytokine storm. So this is really quite a nice description of the sequence of events where the epithelium becomes infected, leads to viral replication. Viral replication results in epithelial damage, release of a variety of chemokines and cytokines and alarmins, which bring in the T cells and the neutrophils and other inflammatory cells through a variety of cytokines. That triggers the cytokine storm, as it's called, and then we have a whole variety then of different circulating markers of that cytokine storm, which can be picked up, such as the various circulating biomarkers of cell activation, and of course the cells themselves. A lymphopenia and an eosinopenia is common, and also an increase in circulating neutrophils, which obviously is the major attacking cell of this dreadful and dangerous condition. As regards the underlying mechanism, why is this virus so effective at getting in to the human compared to the other viruses? Well, it has this unique capacity of paralyzing the first wave of interferon production, the type 1 and type 3 interferons, which are normally fighting the initial viral penetration. And if the virus can obviously trick its way into the epithelium without activating those epithelial cells, then we end up with this very rapidly proliferative cytokine storm. So one of the treatments that we've been involved with, in fact, is giving inhaled interferon to patients when they first get infected with this virus, particularly patients at risk, such as diabetics and severe asthmatics, to see if you can actually replace the missing interferon. That's those clinical trials have just been completed, and we should get some results before too long. But of course, the major problem really is this macrophage, this monocyte macrophage lineage, a very complex cell which has the capacity to morph into this very tissue damaging cell with release of a whole range of different chemokines and cytokines that activates the immune response, and in particular is able to interface with activated T cells and natural killer cells, as well as being able to generate its own cytokines. So most people now believe that the cytokine storm, as it's come to be known as, has its origins in this macrophage with a whole variety of different cytokines that are generated. And here we can see a list of some of those cytokines here, which I shall talk about in a second. Now, the characteristic systemic features of this COVID-19 leads to a variety of organ problems. Outside of the lung, the tissues most frequently affected are the kidney with renal failure, the liver with fatty liver and subsequent liver failure, cardiovascular injury with arrhythmias and shock and hypertension and various forms of cardiac injury, gastrointestinal injury with gastrointestinal symptoms such as diarrhea and vomiting. There's new data suggesting neurological injury as well. And there's obviously coagulation damage, which is the other thing that I talked about earlier on, the microthrombus and the hemorrhage occurring in the lungs. So this is a really very unusual and very profound reaction that occurs in people with a systemic disease. And people have been trying to understand what this systemic disease is all about, because it does resemble to some extent this condition called hemophagocytic lymphohistiocytosis. In other words, the classic macrophage activation syndrome. And this is the so-called cytokine storm following bacterial infection or following very severe injury. And it's where the immune system and the cytokine system become completely activated and leads to multi-organ failure. One of the things about the cytokine storm that occurs in COVID-19, however, is that it's different from the classical MAS SHLH picture in that in COVID-19 it occurs almost entirely in the lungs to begin with before spreading systemically. And it's the lung origin of this that is so peculiar, whereas normally this life-threatening condition begins in the circulation. And shown on the right-hand side here are the various organs that are involved in the cytokine storm, and there's the very central role that the macrophage and monocytes play in its etiopathogenesis. But spreading of this in the lung is the reason why the lung, of course, causes some of the most severe symptoms that these patients experience as they pass through the viral stages. Shown on the right-hand side is an alveolus undergoing this acute inflammatory reaction with neutrophils and macrophages interfacing with type 2 pneumocytes, which were infected with the coronavirus. And you'll see that the communication between that alveolar macrophage and the underlying blood vessel on the right here is a critical one, because pulmonary hypertension due to hypoxemia and due to the microvascular abnormalities is a feature of this disease, which is associated with disease progression. And as with the MAS SHLH syndrome, when seen in other diseases, it's thought that these cytokines, these pleiotropic cytokines and chemokines generated as a result of monocyte and macrophage and T cell activation are the major cause of the subsequent organ failure. And this shows a series of studies that have looked at circulating cytokines in the more systemic severe form of the disease, and those with the blue star where elevated levels of tumor necrosis factor alpha have been found in the circulation, suggesting that TNF may be a major cytokine involved in this. And there have been several studies that have really shown this quite nicely. I'm going to just show two here from Wuhan, which are some of the earlier studies. This one here from Chen et al., published recently on 21 patients, looked at retrospectively who had severe disease, including patients with diabetes and hypertension. You can see in this diagram that TNF alpha is elevated compared to those that had mild to moderate disease, but also a range of other cytokines, including IL-6, IL-8, and IL-10 in the circulation. This is a second study in Wuhan. This is a prospective single-center study looking at 41 patients admitted to hospital with varying disease severity, including those that needed to go onto a ventilator. And again, they were looking at the circulating levels of cytokines. And if we look at this, and I'm sorry you can't see all of the blocks here, but in essence, what it's demonstrating is that compared to those who are healthier or who are mild disease, those that needed oxygen or went onto a ventilator had elevated levels of a wide variety of different cytokines. But tumor necrosis factor alpha, MEP1 alpha, MCP1, IP10, various GCSFs and GM-CSFs, IL-10, IL-7 and IL-2 were all elevated in the circulation, suggesting that this is an important part of the syndrome, this massive release of pleomorphic cytokines. But elevated levels of these cytokines alone hasn't been that helpful in determining which treatment should be used to target which cytokine and when. And this slide is meant to illustrate basically that there's some hierarchy in all of this. And it seems that tumor necrosis factor alpha, interferon gamma, IL-6 and IL-1 beta are cytokines that seem to be particularly associated with very severe disease, requiring people to go on a ventilator, which of course carries the highest mortality. So we've only just got the first results through of a controlled trial of dexamethasone in this disease. This is a very major trial in the UK where 13 to 14% of all patients who had COVID-19 admitted to hospital went on to this trial, and it's called a recovery trial, and they received dexamethasone. And you can see, looking at these Kaplan-Meier plots, that dexamethasone compared to usual care really has had a remarkable effect. It certainly hasn't cured these people, but there's no doubt about it at all that this treatment has definitely ameliorated the aggressive terminal stages of the disease. And therefore, this is now advocated as one of the treatments as one passes from the mild disease into the more severe disease spectrum. Another treatment that's been tried is blocking the interleukin-1 beta pathway. And here they've been using in Italy anakinra, which is the soluble receptor for interleukin-1, which mops up interleukin-1. This was a study in 16 patients in Milan. And you can see, although it's only a small study, again, looking at the Kaplan-Meier plots, that those that received the interleukin-1 beta soluble receptor, shown in red, were better protected than those that had usual care. These are small trials. They really obviously need to be repeated, but it's certainly looking encouraging. Then, of course, I come back to tumor necrosis factor alpha, and here we have a most interesting situation where this macrophage or monocyte, which is circled in red in the center of the alveolus there, is giving rise to large amounts of TNF, which we know also underpins many chronic inflammatory diseases that are characterized by acute flare-ups, such as rheumatoid arthritis and inflammatory bowel disease. So anti-TNF therapy is certainly has a possibility of being useful here, and we recently reported a paper in the Lancet describing why this should be used. And 2 weeks ago in Oxford, a national study of anti-TNF treatment in severe COVID-19 has now been started. So this is another potential therapy that might be of some use. The background to using anti-TNF really is not that we had a lot of experience with this in COVID. In fact, quite the reverse, apart from elevated TNF levels. But the pattern of the disease with the dominant monocyte macrophage activation, the mouse models of various viral diseases that have been investigated using anti-TNF treatment, and in patients with inflammatory bowel disease receiving anti-TNF alone and infected accidentally, obviously, with this virus, seem to be relatively protected against the virus compared to those not receiving anti-TNF. I mean, it's all circumstantial evidence, but nevertheless, it looks as if it might actually be something well worth trying. And then the safety side, well, this just lists a few of the reasons why we feel anti-TNF might actually be reasonably safe or safe in this condition. And again, you can see that its safety profile has been well tried in a whole variety of chronic flaring inflammatory diseases such as rheumatoid arthritis, inflammatory bowel disease, etc. So my last slide really relates to what's going to happen next. Obviously, we want this virus to be eliminated, and vaccines are being tried now in the United States, in South America, in the United Kingdom, and elsewhere around the world, and hopefully one will be found soon. But the key to this really is what happens to the damaged lung in patients who have survived this condition. And you can see on the left upper panel here that the disease usually lasts about 2 to 3 weeks, but even at the end of a month and a half here, there are some patients who are still symptomatic. And the sort of symptoms these people are experiencing are breathlessness, fatigue, lethargy, headaches, just not feeling well. And recent studies now of the original SARS virus lung disease and some very recent studies on COVID-19 are showing that in patients who develop the disease, they develop this florid pneumonic disease, But as they go through, this is day 15 here, you can see that some of the areas of the lung look as if they're becoming organized. And therefore, the acute pandemic phase of the disease, which is the one we're witnessing at the moment, might well, in a small number of patients, but a critical number of patients, pass into either death, which we know about, especially in older people, but also beyond full recovery, a group of patients are going to experience some chronic lung fibrotic disorders and have permanent lung injury. And this is a potentially very serious long-term problem, because obviously if this happens, we do not have good treatments for lung fibrosis, and therefore these patients may run into problems in the long term. So, ladies and gentlemen, that really comes to the end of my talk. I hope you feel I've covered the territory reasonably well. There are probably more unknowns than there are knowns about this virus still, but progress is being made. I think all of us hope and pray that a vaccine will soon become available that does protect the population, so that we can look back on this period as one which won't be revisiting us again, hopefully. Thank you.
Yes, Professor Holgate, thank you very much for that very enlightening talk. Maybe I should just share with you some of the experience that we've had in Singapore, in particular using immunomodulators. At the beginning of the outbreak, sometime in February, March, in fact, one of the combination therapies that we used in the initial lot of patients was a combination of Kaletra, which comprises ritonavir, lopinavir, together with subcutaneous interferon beta. And that was based on some of the in vitro studies on MERS-CoV a couple of years back. What are your thoughts of using subcutaneous versus inhaled beta interferon? Because I think recently a Hong Kong paper also came up with a combination therapy, a triple therapy, which also included ribavirin in the subset of severe COVID patients who they treated.
Yeah. Well, I think myself that giving any drug systemically will maximally treat disease processes which are systemic. But I think in terms of interferon, if you want to prevent the viral replication very early on, and this virus evades the first step of the primary innate immune response, then you need to give it high concentrations locally, really. Just like with a beta agonist or a corticosteroid, if you give high concentrations in asthma of those drugs, you get really high responses locally without the systemic response. But as I said in the talk, and you're quite right, Bernard, that this does pass into the system, and I'm quite certain that systemic drugs are going to be needed for that. But I think if we can get to the patients early in the risk groups, the diabetics and those with chronic severe disease, and give them high local concentrations, that might work better. I don't know the answer because those trials haven't been completed yet. With the combination therapy, the recovery trial, which is this massive, large NHS trial that's being done, has included the antivirals in that trial. And of course, they haven't all read out yet, and they're all still reading out. The dexamethasone was the first one to read out. But they're putting thousands of people into these trials. And so therefore, because they are umbrella trials where any good treatment that's been shown to work even in other diseases are being tried, and we won't know the answers to this probably for another month or two yet. So I'm quite convinced that combination therapy will be the way forward. But the big question right now is which combination and when, because this disease passes through these different phases, and one treatment that may be active at one phase may not be active at another.
Yeah, so in Singapore, we also went through a phase of giving off-label tocilizumab. And one of the reasons why we had to give it off-label was because we didn't have access to some of the anti-IL-6 inhibitor trials. From our experience, we find that it's a little bit difficult to determine the window of opportunity when patients would. So what we tried to do was to try and identify the patients when we became hypoxemic before they ended up intubated. And it was a little hit and miss. Some patients did well, some didn't do well because of comorbidity. So it's quite hard to find a good biomarker to decide who are the patients who would benefit, I find.
Yeah. Well, I mean, there's a lot to learn, as you correctly point out, Bernard, and until we actually know more about the cytokine hierarchy in these different stages of the disease, knowing exactly which one to target when is going to be the key thing, I think.
Yeah. Turning a little bit to another topic, which is asthma, I think there was a recent publication from the UK about asthma exacerbations during COVID-19 versus asthma itself being a risk factor for COVID-19 infections. What's the experience been like in the UK?
Yeah, well, I tell you, you're right. Well, certainly during the lockdown period, the 3 or 4 weeks we've been not communicating as we normally do with between humans, virus exacerbations have dropped dramatically. So hospital visits, demands for pharmacy use of emergency treatment has dropped. And probably the most likely reason for that is the fact that viruses are not being handed around, because we know that viruses are a major cause of asthma exacerbations. Also, air pollution has dropped quite dramatically in this country as a result of this lockdown and vehicles. And again, we know asthma exacerbations are linked to increased air pollution. So those are two reasons that might together contribute to reduced exacerbations. I think, as far as I can tell, the Chinese experience, and I think we're getting the same here, that mild to moderate asthma are relatively protected against COVID-19 compared to severe asthmatics who seem to develop really quite a nasty form of the disease. So again, it may be the fact they're taking inhaled steroids on a regular basis in moderate to mild asthma that's protecting them in those early stages of COVID disease. But again, I'm afraid it's speculation because we don't know for sure. I'm sure all of this will be found out in time, but it is interesting that asthmatics haven't got a lot more disease than people anticipated in the first instance, and it's only those with very severe disease that have been affected.
Okay, so my question, Professor Holgate, is you showed this data showing the macrophage-related inflammatory cytokines like IL-6 and IL-10 and TNF-alpha in severe versus moderate cases, and also highlighted the importance of TNF-alpha. So I was just wondering, since you also talked about combination therapies, do you think there's a placement for a combination of anti-IL-6 and anti-TNF-alpha as a treatment that at the early stages when you or is there a cutoff mark for the levels of IL-6 and TNF-alpha that we would say at this stage we would give anti-IL-6 and anti-TNF-alpha?
Yeah, well, I think so. I mean, it's almost a replay of the rheumatoid arthritis, inflammatory bowel disease story, isn't it? Because obviously those two cytokines are involved in those disorders. And it seems that the way they're managing arthritis is that they give one, and then if that doesn't work, they then give the second one. But combination therapy hasn't become a routine, possibly because of expense apart from anything else. So I don't know is the answer, Ruby, whether there's going to be TNF, type disease more than IL-6 type disease, more than IL-1 beta, or whether in fact all patients are going to benefit from having more than one cytokine blocked. And I just don't know the answer to that, I'm afraid. It's something we will eventually know, I'm sure, but the problem is these drugs are enormously expensive, and if large numbers of people need them, there's going to be real problems, I think, in availability. And you can't imagine what would happen in countries like Africa and so on, where these drugs just aren't available, really.
Yeah, and in relation to this, I was also thinking that because several of these cytokines, like IL-6 or TNF-alpha, the epithelial cells are an important source of these cytokines, besides the macrophages. I mean, I'm sure it's just speculation at this stage, but would you speculate, because now that we know that how important, and your department and your group has shown that so much, the importance of the epithelial barrier dysfunction in airways disease, especially asthma. Do you think that there could be some way of actually preventing or dampening the effect of the severity by increasing the epithelial cell integrity and reducing the cytokine storm from the source of the epithelial point of view?
Yeah. Well, of course, the virus has got to get from the nose down to the alveoli, and it infects preferentially the type 2 pneumocyte, which only make up 5% of the pneumocytes in the lung anyway. And so the journey from the nose to the alveoli, it takes a finite time. And what's strange about this virus is that patients can be asymptomatic and carrying the virus and spreading the virus before actually they develop, symptoms that tell them they've got a problem. And of course, this has been the big issue of its transmission, really. And children, likewise. I mean, there's a big conundrum around children as to whether they can be super spreaders in a way or not, because they're not symptomatic, but they're also infected. So, what is the journey? How long does it take? Could you put something in the upper airways that prevent the virus from traveling down? Well, I think that's certainly possible. I mean, the virus has got to replicate. It then has to kill the cell to release its, virions so that it can be inhaled further down the lung. This all takes time. So it's, I think you may be right. If you could increase the resilience of the epithelium, as obviously children have an epithelium that's more resilient, then obviously the progression of the disease might be ameliorated. And that would be a very, useful thing to know. But until we know more about the transmission down the respiratory tract and exactly what happens as it moves down, I think this, we don't have anything to offer as yet. I think they may come, and I think what's kind of sad about this whole story really is that we've had 3 viruses now in close succession, relatively, plus a whole bundle of influenza viruses, and yet we still haven't got the resilience of the innate immunity worked out to a point where we can protect the lung against all these viruses, which I think in the end would be what would be wonderful to achieve. Because what we're doing at the moment is picking one virus off at a time. With vaccines, well, we know that that's a real problem with RSV, for example, or with the different influenza viruses, because people the viruses mutate and you get a different form. So the innate immunity is important. And in this respect, vitamin D, for example, I think it's an important agent to make sure there are adequate levels, because that increases the innate immunity in the lung and increases epithelial resistance to virus infection. I don't think chloroquine does, though.
Except, okay. The other thing in close relation to this is like, clinically people are looking at levels of D-dimers, level of IL-6, level of ferritin, several markers, CRP and all. So is there, would you speculate as if we had to put all these together, would there be 1 or 2 markers that would actually predict somebody who comes in hypoxic, but maybe that person is more likely to require an NIV as compared to someone else who won't require that? Is there some indication, at least, at this stage?
Yeah, I think so. And that work is actually being done. And I think day by day, we're seeing proteomic papers coming out, metabolomic papers, a variety of different pan-cytokine papers, and sort of using artificial intelligence then to try and put together a story like you're suggesting, where you can get a hierarchy of cytokines which dictate the disease subtype and what therapy you should be introducing at that stage, is exactly where we should be taking this. And I think that will definitely happen. it hasn't happened yet, Ruby, and most people are just bunching these biomarkers together and not attaching the discrimination yet that they probably deserve. I mean, they're able to separate now those that go on to need ventilators from those that don't, but that's a pretty crude cutoff, really, when you think about the biology of this condition. So I think there's quite a bit to go. I think within the next month, we're probably going to have some really quite interesting proteomic data, where there'll be 4 or 5 biomarkers for different stages of each disease, which will help then in the categorization of the disease process.
Very much. There's a question that I think you've already answered, because I talked about I wanted to ask about the potential risk of mutation of the virus by the time we have the vaccine, which is it's something that you already said. That's always a risk, right?
Yeah, well, of course, and this virus is mutating. I think there are about 22 mutations have been tracked since the original virus was studied in Wuhan. But that's it's compared to influenza or the common cold virus, the mutations don't seem to be increasing its virulence, which is the important thing. But there are 400 different coronaviruses in bats. I mean, and of course, how much do we know about those? So it may not be the same. It may be another one that gets out. So, I mean, we need to understand, and I think the United States and China are doing this, building up an atlas of the virome, really, so that we can, of all the different coronaviruses and different types of respiratory virus, so we can learn where the commonalities and differences are. And I think once we've got that, then vaccines can be targeted to particular components that may protect against more than one virus subtype. But all of that is being done now as a result of this pandemic. But you could say it's a bit late, but never mind, it's being done.
Yeah, you touched upon the point of the viral virulence, and that took me to this concept that Sweden had adopted, which has actually failed, of the herd immunity. So do you have any comments on that?
I mean, they tried to introduce that here initially, and it went down like a lead balloon, I can tell you. I mean, currently, the surveys that we've done in the UK using the antibody tests show that between 5 and 8% of the population have been infected. Well, to get herd immunity, you need 65% of the population to be infected. We're nowhere near. I mean, just nowhere near. Now, I don't know what the infectivity of the population is like in Singapore, for example, or in Japan. I mean, it may be that more of your population have had mild disease and have been infected, but until we have the antibody test, it's impossible to be able to find that. Bernard, have you got any data in Singapore?
Well, I think in Singapore now, well, we have two portions of the population. One is the community at large, and the other population that we have a bit of a problem trying to control currently is still the foreign workers who live inside the dormitories. So currently we still have about 200, 300 cases who are being isolated and diagnosed in that population. So the only good thing about that population is that it's a pretty young migrant population. So the disease that we've had has been fairly mild throughout. So what we've been doing in Singapore in terms of serological testing is that we have used a combination of nasopharyngeal throat swabs, PCRs, with serological testing to try and determine really whether a lot of these migrant populations have already gone through the acute infection phase, and now whether they are like 2-3 weeks down the road. And we use that as a strategy to try and isolate them and create clean dorms to put the migrant workers who have not been infected into those dormitories. And for those who have been who are infected, then what they do is they go into another cluster of dormitories, basically or community care facilities to recover. So that's how we're using a combination of PCR testing as well as serologies.
Right, excellent. And what proportion of the immigrant workers are antibody positive?
The antibody positivity varies depending on which dormitories there were and whether there were large outbreaks. So in some of the bigger dormitories where we saw the earlier phases of the outbreak, we had almost as close to 80 to 90% of the workers there who had already seroconverted.
Right.
Whereas in some of the other smaller dormitories, the seroconversion rate was quite low. So we assume that those were probably the clean dorms where there were not a lot of infection which was going on within those dormitories. So we use that as a way to segregate the populations in the dormitories.
And in Japan, actually, we started testing pretty late, but mainly we do the RT-PCR. We don't do the antibody testing in Japan. It's not commonly done. And the only good thing I would like, I think, I would presume why we're not in a state like maybe Italy or UK or America was we started the face masks, and which is a very common thing in Japan in the pollen season. So we started the face masks very early. We started the hand hygiene and general level of hygiene very strictly right in the beginning. And we also started a certain amount of social distancing, not really that much, but telework had already started before they did the lockdown. We didn't have a very strong lockdown, because there is no law that allows a strong lockdown in Japan. And other thing, what they did is they closed the schools pretty early. So these were the things. But what I've always wondered is when I look at India and when I look at Japan, the two things. One is or Philippines. We talk about, and Singapore, we talk about high temperatures and the virus doesn't survive. The virus is surviving like anything in these high temperatures. The other thing is we're talking about the elderly population and the younger population. When we look at India, we have a lot of younger population, and here in Japan, we have the elderly population. So sometimes I don't know what the reason is. Is it something genetic? Is it the food habits here, which has plenty of vitamin D naturally in their food? Or is it generally the hygiene and then the fact that in Japan, we don't have a real system of physical contact between people. The people always maintain distance. So is that the thing, or is it a different strain that we have here? I think these are open-ended questions that would be interesting to know.
Indeed, so more unknowns than knowns.
Actually.
Yeah, so I think as in Singapore and the rest of the world, everybody's wanting to travel, everyone wanting to open the economy. So we've opened the phase 2 about 2 weeks already. We're still trepidating whether there will be a second wave or not, because if you look at the general population, the way people are doing social distancing and the way people are not wearing their surgical masks properly is a little bit of a concern. And I'm sure that would be the same elsewhere in the world too.
I'm sure you're right. Yes, I'm sure you're absolutely right. So we'll just have to see, won't we?
Yeah. Anything else, Bernard? Bernard? Any more questions?
I think we've had a very good discussion, and we've had a very good sharing of our different circumstances in the different parts of the world. So let's see what the science, the technology, as well as the policies will bring in the next few months. Hopefully this pandemic will end sometime soon.
Yeah, indeed so. I hope so. Yeah, we're all wishing that. We've got a winter, and it'll be a winter of discontent if it does not end soon.
Well, just one last question, Steve. Any idea about Professor Holgate? Any idea about when it's likely? Because I think there are about 3 vaccines now, like ahead of the other. Although many more are being tried, like 3 vaccines are like ahead of the others. What do you have any take on that? I mean,
no, not really, Ruby. They're all very different, which is probably a good thing. Using different technologies and taking advantage of different parts of the immune system. No, I mean, you know, how long does it take to generate a vaccine? Well, you take something like RSV, we still haven't got one. And yet, you know, that's been going on for 40 years. And you ask yourself, well, why not? If the, you know, so that I think, you know, even in theory, it should be possible to get one and all the rest of it. Whether we actually end up with one that's going to be sufficiently efficacious in protecting a whole population, I think we just don't know. It may take years. I mean, it's the science behind vaccinology is fine in terms of molecular targets, but not so good in terms of whether that does the job when people are given it systemically, you know, across a broad population. And you know, that's true of all vaccines. And yeah, so very difficult. In a way, you know, an attenuated vaccine would probably be better, which is multivalent rather than going for single target proteins, because we honestly don't know that targeting the spike protein, for example, or whatever, you know, is that powerful enough to be able to block this? I don't know. Yeah, so there's a lot of speculation, a lot of potential, but nobody knows the answer really.
Thank you very much. So Professor Holgate, thank you for a brilliant talk as always. And I'm sure our membership are going to be extremely excited and thrilled and will learn a lot from this talk. We have learned a lot. I thank my co-chair, Professor Bernard Thong, joining us in this very important webinar, and wishing everybody stay safe and well, and hope we can get over this crisis very soon and meet face to face.
Indeed so. Thank you very much. And thank you, Bernard, and thank you, Ruby. It's been an absolute pleasure.
Thank you, Stephen. Have a good time.